Welcome to this comprehensive training on ICH Q3A and Q3B, impurities in new drug substances and drug products.
I'm your presenter. Today we'll cover the key principles, thresholds, and practical steps to control impurities.
Q3A and Q3B were adopted in the mid-1990s and revised in the 2000s to harmonize impurity control globally.
The International Council for Harmonization creates guidelines to harmonize pharmaceutical quality, including impurity control under Q3A and Q3B. These guidelines ensure patient safety, regulatory clarity, and consistent impurity control worldwide. Quick recap: Q3A focuses on drug substances, Q3B focuses on drug products. Next, we dive into Q3A: scope, impurity types, thresholds, and analytical requirements. We'll keep this practical: thresholds, decision trees, and how to demonstrate safety.
By the end, you'll be able to identify thresholds, design analytical strategies, and apply Q3A/Q3B in development.
Q3A applies to chemically synthesized new drug substances, including some peptides and oligonucleotides produced chemically. It does not apply to biological products or proteins isolated from biological sources. Organic impurities: starting materials, intermediates, byproducts, degradation products, reagents, and catalysts. Inorganic impurities include metals and salts. Residual solvents are covered by Q3C. Critical concept: thresholds based on maximum daily dose determine identification and qualification requirements. For doses, 2 grams per day. Identification threshold 0.10%. Qualification threshold 0.15%. For doses above 2 grams per day, both identification and qualification thresholds are 0.05%. If impurity, identification threshold, identify it. If qualification threshold, also demonstrate safety. Q3A requires validated analytical methods that detect impurities at threshold levels and are stability indicating. Methods must be specific, quantitative, and sensitive at threshold levels. In practice, chromatography with orthogonal techniques for identification, MS/NMR, is standard for characterization. Q3B extends impurity control to final drug products. The patient-facing dosage form applies to chemically synthesized drug products, immediate and modified release, intended for marketing. Sources of product impurities include carried-over substance impurities, manufacturing form degradants, storage degradation, and interactions with excipients. Drug substance-related, drug product-related, and interaction products with excipients. Q3B thresholds differ. For doses, 1-day identification, 0.10%. Qualification, 0.20%. For doses above 1 gram per day, identification 0.05%, qualification 0.20%. During stability studies, any degradant that rises above thresholds must be identified and qualified. Q3B thus links impurity control to life cycle stability data, shelf life, storage conditions, and possible formulation changes. Remember, Q3A controls substance impurities. Q3B monitors what happens after formulation and during storage. Cross-functional review of stability and impurity trends is essential for regulatory readiness. Start with risk assessment. Review synthesis, assess degradation pathways, evaluate excipient interactions, and manufacturing variables. Analytical methods must separate and quantify impurities at threshold levels. Develop robust, stability-indicating methods. Use orthogonal techniques for identification. HPLC, HRMs, NMR, validate LOT/LOC at or below thresholds. Include method validation reports and impurity specifications in regulatory dossiers. Essential docs, impurity profiles, method validation, safety qualification data, stability trends, and risk controls. ICH principles are global, but regional submission details vary. Check FDA, EMA, PMDA specifics. Complementary guidelines: Q3C for residual solvents and Q3D for elemental impurities. Consider them in your control strategy. Keep abreast of emerging analytics, QBD, and risk-based approaches in regulatory reviews. Ensure cross-functional communication. Chemistry, analytical, regulatory, toxicology, and manufacturing must align. Control starting materials via supplier qualification and robust change control to avoid unexpected impurities. Case 1. Drug substance. Max daily dose 500 mg. Impurity detected at 0.12%. Dose a 2g day. Identification threshold 0.10. Exceeded, so identify the impurity. Qualification threshold 0.15 not exceeded. No safety studies required. Action. Identify the impurity structure and include it in the specification. Case 2. A degradant reaches 0.25% at 24 months. Identification threshold exceeded. Qualification threshold 0.20 also exceeded. Identify, qualify, and consider shelf life or storage changes. Possible responses: identify degradant, perform toxicology qualification, or tighten storage and labeling. Common mistakes: wrong dose for thresholds, incomplete identification, inadequate method validation. Best practices: early integration, stress testing, robust methods, supplier control, continuous monitoring. Implement via risk assessments, validated analytics, and strong quality systems. Now a quick look ahead at advanced analytics and regulatory trends. Emerging tools: high-resolution MS, 2D chromatography, real-time monitoring, and AI-assisted method development. Regulatory trends: risk-based approaches, QBD integration, lifecycle management, and harmonization efforts. Key points: Q3A/Q3B provide harmonized impurity controls. Thresholds are exposure-based. Risk assessment and analytics are essential. Success factors: early planning, robust methods, cross-functional collaboration, proactive impurity control. Quick check. 1. What is the qualification threshold for a 500 mg dose? 2. List 3 impurity sources in drug products. 3. When to conduct safety qualification? 4. Required method characteristics? 5. How do stability studies relate to Q3B? References: ICH Q3A R2, Q3B R2, Q3C, Q3D, and regional guidance from FDA, EMA, PMDA. Thank you. Impurity control protects patients and supports regulatory success. Apply these principles across your programs.